So I'd like to start. I'd like to thank all of you for coming out today, making your way to this remote part of campus for many of you. And I'd like to thank the Stork Committee for being brave enough to try one of these in the summer. And I'm pleased to have quite a good turnout. It's my great pleasure today to introduce Dr. Bo Barker-Jørgensen, today's seminar speaker. But first I want to briefly thank the Storr Endowment for sponsoring this lectureship and their foresight, Tracy and Ruth Storr's foresight in funding this endowment will become increasingly important as we go into continued cost-cutting measures. At least we'll be able to hear world-class seminar speakers still, regardless of what else they cut out of our budgets. Bo Barker-Jørgensen received his undergraduate degree in zoology from the University of Copenhagen, and a professor at the university at the time, Tom Fenchel, had been asked to take a professorship at Aarhus on the mainland and asked Bo if he would accompany him as his first graduate student. Bo did. He stayed on after his PhD at Aarhus as a lecturer and as a senior lecturer, and finally in a special Danish Natural Sciences Research Council lectureship. Henry Blackburn was the chair of the Department of Ecology and Genetics at the time, and during the 80s and the early 90s, the group there, which included Bo, Niels Peter Revsbeck, Jan Sorensen, was a spectacular place to go and learn about nitrogen cycle, sulfur cycle processes in sediments. They self-deprecatingly called themselves the mud group, but it was a real area of wonderful science and lots of innovation. In 1992, Bo was asked by the Max Planck Society to become a co-founding director of a Max Planck Institute for Marine Microbiology in Bremen, which he did, and he continues to share that directorship with Fritz Vittel and Rudy Amon. 2 years ago, he was asked to open up a new center for geomicrobiology that was jointly funded by the Danes and the Germans, and this would be coming back home to Aarhus. And so he is wearing both hats still, but gradually transitioning back to Denmark. He's been an extraordinarily productive scientist on a wide range of microbial and geochemical topics. His peer-reviewed publication list runs to about 250 plus publications, including 21 in Nature or in Science. Much of his focus has been on both the reductive and the oxidative part of the sulfur cycle in marine sediments. And because sulfate is such an abundant ion there, these cycles are extremely vigorous. I won't go through a list of all of his major accomplishments because it would take away most of the time he has for the seminar. Most recently, his thoughts and scientific attention have turned to microbial processes at the limit of microbial life. He once convinced a graduate student to try to culture and understand sulfate-reducing bacteria living at 0 degrees C or even sub-0 degrees C temperatures. Once the student, getting a bit nervous because he was now a year and a half into a 3-year project and had just achieved pure cultures, found out that these organisms could double at best in a week. The student was a bit nervous but still finished the project. Bo is delving deeper into the sediments, into slower and slower microbial processes, but the payoff is that this deep subsurface biosphere that he's now beginning at this new Institute for Geomicrobiology to investigate, the payoff is that the microbial populations there, by some estimates, may be as large as all of the biotic carbon in plants in the biosphere, and that biotic nitrogen and phosphorus in these environments may be 10 times that contained in plants in the biosphere. So today's topic is here for you to see, and I'm extremely pleased to introduce Professor Bo Barker-Jørgensen.
Thank you very much for my introduction. bring you into a new environment that is probably very strange to most of you, that has been discovered within the past 20 years. It's the deep subsurface. And to me, it has been like discovering a new continent full of organisms we have never met before, for which we don't know how they can make a living, but where we realize that this is a continent teeming of life. And I've called it with the subtitle The Starving Majority, and I think at the end of the talk you will realize who I am. So for those of you who are not in marine sciences, just as a stage, you know about plate tectonics, and you should find out about it. You know that there's fresh basalt being generated at the mid-oceanic ridges, pushed out to the sides, and the Atlantic pushed continents away. In the Pacific Ocean, it subducts under the continents on both sides. And it takes tens of millions of years for the sediment crust to glide, the ocean crust to glide out to the sides. And that means that over these tens of millions of years, there's a continuous deposition of planktonic debris, carbonate shells, plastic material from the continents, from the sediment. So the sediment fills up, and you may have hundreds of meters of sediment at the end piled up on the crust. And if you drill down through the sediment, you drill down through ocean history over tens of millions of years. In this part of the ocean, if you go back a bit more than 5 million years, that was the time when the Central America was closed up and closed the connection between the Atlantic Ocean and the Pacific Ocean. So there have been dramatic shifts over the time that you look into in such sediment cores. And over the past 20 years, there has been ocean drilling which has brought up sediment cores that have been used for counting of bacteria. Now, the research vessel here, JODIS Resolution, is still in operation as the main operating platform for gaining these samples. The sediment cores are taken with normal oil drilling coring technique, but there is a piston core that shoots out ahead of the drill bit, and when you take out sediment cores from that 10-meter long core liner, it is in fact possible to get non-contaminated sediment samples. It's hard to believe when you look at the sterile technique of the drilling crew that this is possible, but you can check whether there is contamination. We continuously pump tracer perfluorocarbon into the pumping water, and if we can detect that this has penetrated into the samples, we do not use it for microbiology. So over the years, we have great confidence that we know when there is contamination. The most recent drill ship is this of Chikyo of the Japanese. It's twice as high, 120 meters high drill tower. It's a huge operation, and of course this is altogether a very expensive research operation, mostly for geosciences, but now also with the deep biosphere research as one of the 3 major emphases in the program. And over the years, especially the group of John Parks in Bristol, now at Cardiff in Britain, have been obtaining samples and done simple microscopic counts after fluorescence staining of DNA in the cells. And this is the database as it looked in 2000, and today it looks just about the same. There are more red dots. And what you see here is a double log plot of the total number of microbial bacterial-sized cells in sediment cores, going from the surface here 1 centimeter, 1 meter, 10, 100, 1,000 meter depth down into the sediment. And then you see here the log of the bacterial numbers. There are between 10 to the 9 to 10 to the 10th per cubic centimeter up at the sediment surface, dropping in a log-log linear mode down to about 10 to the 6th or 10 to the 5th at a sediment depth of 1,000, a kilometer depth below seafloor. So there are bacterial cells all the way, and this enumeration has gone on. Still, there are new records set. The latest one published in Science last year is from the Newfoundland margin. You know, the Atlantic margin is a passive margin, so sediment gets very old and very thick. at 1.6 kilometers depth in the sediment, it had an age of 111 million years, and in that it had the temperature between 60 and 100 degrees, and there were plenty of cells still to be counted down there. So it seems that everywhere you really look, you will find bacterial cells. So we can conclude generally that in these deep deposits of organic material over geological time, there are bacteria, there are archaea that are living there. Generally, they all live under high pressure, more than 400 bar pressure. And so far, the only limit that has been seen to their distribution is temperature. When temperature goes above 100 degrees, you do not find bacterial cells anymore. And it seems that above that temperature, when the sediment gets buried below temperature limit of 100 degrees, it gets sterilized. There's some examples of that that the oil industry is very interested in. If you have a deeply buried sediment that has later been uplifted and now has crude oil, it seems that this oil is less degraded because it does not have a viable bacterial population. So there are bacteria all the way in the sediment, and why is this interesting? Well, for one thing, the marine sediments are the largest carbon reservoir on Earth. The buried organic carbon in the sediment is such an important part of the overall global carbon cycle. This is how CO2 gets sequestered in the long geological term, and the efficiency with which microorganisms are able to mineralize the organic carbon back to CO2, release nutrients, sets the balance between burial and release of CO2 in sediments. And in the long term, that also affects the general chemistry of the ocean. It affects also oxygen levels in the atmosphere. So when you look at the distribution of bacteria in the sediment and then consider how thick are these deposits, you can start to make a global estimate of what is the total number of bacterial cells in the ocean seabed. And the thickness of marine sediments vary a lot from these young sediments in the middle of the oceans that are only tens of meters thick, and then to the thickest that are some 10 kilometers or even more. And there was, about 10 years ago, a bold attempt by Barney Whitman and co-workers to estimate the total global biomass and numbers of prokaryotic cells in different environments. And they compiled all they could from the literature and came to the astonishing conclusion, which they published in PNAS in '98, that only a small fraction of all prokaryotic cells are on the surface of Earth. 3 to 4% of them are in the ocean, another 5-6% are on land, and the rest are in the deep subsurface. Half of them in the seabed and the other half in the terrestrial subsurface. So when you consider that by far the largest fraction of all prokaryotes, that is more than 90%, are in the subsurface, then you wonder, well, how much of the living biomass on Earth may that be? And although prokaryotic cells are extremely small, the estimate by Whitman was that between 1/10 and 1/3 of all living biomass on Earth is in the form of prokaryotic cells in the subsurface. This was hard to believe. It still is. And so we have tried to test this in different ways. Do these numbers hold? Is this really a realistic estimate? And one thing you observe in this database by John Parks and his group is that it is sampled wherever this ocean drilling program has been coring. And since this is run mostly by geoscientists, they have been going in geologically interesting and active sites, and this is mostly at the ocean margins. So most data have come out of the ocean margins where there's higher productivity, higher organic sedimentation, and for that reason, probably more microorganisms. If you go into the ocean gyres here in the South Pacific, where there is very low chlorophyll, as you see in the color code here, or the northern gyre, then it's a different matter. There's very little sedimentation. You can have sedimentation over tens of millions of years of a few tens of meters. And if you look at the mineralization rate measured as the respiration by the bacteria, you can model that by measuring oxygen penetration into the seabed. And the red data points here are just about to be published now. The first data that show that in such parts of the ocean, oxygen penetrates tens of meters down. The blue line is then the model extrapolation down to here, where it hits the crust, which is in depths of between 6 and 70 meters. And that means in this part of the ocean, oxygen penetrates all the way down to the ocean crust. We have recent data from the northern gyre here, from a cruise this year with piston cores taken down to 30-meter depth in the sediment, and there was oxygen all the way down in those. So these are extremely low in organic mineralization rates. And if you count bacteria there, well, it's difficult because there are so few bacteria that you cannot, in the microscope, just smear out sediment after you stain it and then find a bacterium. So you have to extract the bacteria from the sediment and then count them, and you lose a fraction of them. But this is the first data set that show from that South Pacific gyre, and it shows numbers that are 2 to 3 orders of magnitude below the main database that has been used by Whitman and co-workers. So if you go into those parts of the ocean, you have lower numbers. Now, one important thing with such a database is that when you look at it in a log-log fashion, it seems that data scatter nicely around the linear correlation line. But in reality, when you try to extrapolate to the total number of bacterial cells, you cannot use a log-log linear correlation. You have to do an arithmetic calculation in which the high numbers count much more than the low numbers. In reality, if you do a linear, log-log linear calculation of the total number of cells, you get a number which is at least 5-fold lower than what you get if you do an arithmetic calculation. I did that recently with John Park's original data. So there are factors that will lead to lower numbers, there are factors that will lead to higher numbers, and in balance, Whitman et al. May not be very much off the real numbers. But the database is building up, and we will know more in the coming years. But it means that really most of prokaryotic life exists in the deep subsurface. And that means under very low energy flux down there, under high pressure. So most microbiology occurs under more than 400 bars of pressure, for which we do not know all that much of the adaptation. With these numbers by Whitman and co-workers, one would really wish that we had other means of estimating the total biomass. And this is the last I will say about total biomass. But there is another way. And this is a quantification of the intact polar lipids in the membranes of bacteria and archaea. So you can quantify the total amount of these intact lipids. After the cell dies, they hydrolyze, you have the individual polar lipids, but in the intact form, they are generally in the live cells. And if you quantify those in different parts of the ocean, you can again build up a global estimate, which was done in a publication last year by Lip and co-workers. And they came up, well, you calculate the total amount of bacterial membrane, basically. And then you have to make the assumption that all these bacteria are spherical, half a micron of size, which is sort of a mean size that in the counts of John Parks and his group. And then you can estimate the numbers of prokaryotes. And the number they came up with, astonishingly, was within a factor 2 of the estimate of Whitman et al., which is a bit coincidental when you look at the logarithmic scatter around the mean here, but still, so far there's nothing to indicate that Whitman et al. Were wrong. So we have to realize that we are dealing with an important part of life in the subsurface. Then the next question, of course, is that, okay, you can find these cells. They have intact membranes. They are not dead. Or you could argue, well, perhaps they are really dead and they refuse to go away because there are no living bacteria around to degrade them. But I'll give you a few examples why we think that is not the case. One thing is that you can do in situ hybridization of these cells. So you can hybridize ribosomes that are apparently in sufficient number in the cells and sufficiently intact that the normal probes that we use for hybridization function. And you can do hybridization of total bacteria, of total archaea. And here's one example of the result of that from one core that was taken in the eastern tropical Pacific, going down from the sediment surface to 400 meter below surface. And the open circle shows the direct microscopic count of total cells. And then the closed squares shows the number of cells that could be identified by fish fluorescence in situ hybridization. That was between 10 and 30% or even more of all the cells. And if we take the intact membrane, if we take the occurrence of intact ribosomes in the cell as an indication that they are not dead, they should be live, whether they're in a dormant stage or not, we don't know, but at least they're live cells. So the other main question, of course, is who are they? And the most basic one is to which domain do they belong? Are they bacteria or are they archaea? And here's another study doing quantitative PCR of 16S ribosomal RNA genes in bacteria and in archaea. And this is now the black squares of the total prokaryotic cells down through this 250-70 meter sediment core. And you can see that the quantification of bacteria based on 16S genes yields about the same number of cells as the total direct counts, which would indicate, well, most of these cells are bacteria. And the archaea seem to be much lower. So this was published, and I was on that publication 2005. A year later, there was another publication, partly based on fluorescence in situ hybridization, partly based on quantification of the intact polar lipids, either lipids from archaea, ester lipids from bacteria that could distinguish the two groups. And the conclusion there was that most of the cells were archaea. So this was somewhat embarrassing that we were not even agreeing on which domain was dominating in the subsurface. And one of the problems that we know now as the database on 16S sequences is rapidly increasing, partly based on high throughput sequencing effort within the past year or two, is that we did not know sequences to a large extent when this work was done. And the primers used for quantifying the archaea had many mismatches with archaeal sequences in the subsurface. So this is an underestimate. And recently there has been an intercalibration where the group of Kai Hinrichs in Bremen has then prepared deep subsurface samples, amended some of them with bacteria, some with archaea, sent out to all the groups in the world, working with these quantifications, and we are still eagerly waiting for the results to be published. Has not come out yet. And some people will be happy about the results, others will be very embarrassed. And I will not say who belongs to which group. But there has been quantification through the intact polar lipids of archaea versus bacteria that has indicated, this was published last year in Nature, that only in the top 10 centimeter of these sediments that are compiled here do we have a dominance of bacteria, and below that it's the world of archaea to a large extent. Now, there's more to it than that. It may be that archaeal IPLs are better preserved than bacterial IPLs, so We are still waiting for this intercalibration. But I think the result will show that the truth is somewhere in between. You have both archaea and bacteria in great abundance. What this shows, though, is that we have really a need to understand who are the archaea in the subsurface. And this is then a big problem, because it turns out that we have here all novel lineages of archaea. There are so many sequences coming out now in 16S, and they're all new. They have been grouped and given names, mostly based on where they were first found. So you have marine benthic group B, which is very dominant. There's the miscellaneous crane-archaeoidal group, which already tells you a bit about that this is a mixed bag of organisms of which we know very little. South African gold mine urearchaeal group. You can guess where this was first found. Unfortunately, you can also find it other places, also in the seabed. Marine benthic groups. So common to all these groups is that there is not one single cultivated representative of those major phylogenetic lineages. There is one of the groups, marine group 1 archaea, where there was the first isolate a couple of years ago, which was an aerobic autotrophic ammonium oxidizer from the deep sea, from the water column. And we were very happy about that, but this is really not moving. People are hunting for these organisms, trying in enrichments to find these sequences, but it is not moving at the moment. I think we do not have the right approaches currently. There was a recent compilation by Andreas Teske of all these 16S sequences from the archaea, and what you should see here in this phylogenetic tree is basically that you have two major groups of archaea, the urearchaeota over here, and what you have over in this branch are all the cultivated urearchaeota, and all the rest are non-cultivated lineages that branch just as deep. Here you have the crane-archaeota, and right here you have all the cultivated crane-archaeota here, and then the rest have no cultivated representatives. And you have even deeper branches here for which we have no representatives. So that means we have an environment here which comprises most of all prokaryotes on Earth, and where we hardly know anything about the function, the physiology of the organisms based on the sequences that we can pull out. So we have a long way to go. One needs to develop new methods, obviously, and I will talk a bit more about that to understand who they are, what they are doing. It's an interesting question. Why are they all archaea, or dominantly archaea? What are properties of archaea that would be favorable in the deep subsurface? And one very important property of the deep subsurface is the extremely low energy flux, extremely little energy available for each cell. And this could be one property. There are a number of energy requirements for a cell to survive and repair DNA, generation of new proteins. One specific property that is different between the archaea and the bacteria is the permeability of the membrane to protons. It's about 100 times higher in bacteria than in archaea, and that means for cells that have a proton motive force, there is a continuous loss of protons and of energy thereby. And this is, I think, one distinguishing property between archaea and bacteria that might be relevant. It may also be that under extremely low energy flux, that proton motive force, sodium motive force may be significant. The permeability for sodium ions is 100-fold lower in the few studies that are available than in bacteria. So one has to start looking for the limiting properties of life at extremely low energy flux. What I had shown you before was all based on 16S genes, and of course one should start also to look for different functional genes. There has been a recent metagenomic study published last year in PNAS by Biddle and co-workers, in which they had extracted DNA from a number of subsurface environments. And one thing they observed was that up to 85% of the genes could not be identified, had no apparent homologies with known genes of known function. They looked into the zone in the subsurface that was dominated by sulfate reduction as the terminal mineralization process, and they could hardly find genes related to sulfate reduction. The same in the methanogenesis zone, hardly find genes related to methanogenesis. So sequences may be very different from what is in the database, and we need new high-throughput sequences efforts to understand the diversity. So much about the diversity down there. So what are the processes? What are they living from? If you can constrain the main geochemistry of the sediments, that is, you have a handle on the input in terms of organic carbon, oxidized, reduced minerals. If you can measure accurately the net changes in the geochemistry of the sediment, then you should be able to constrain also the possibility for energy metabolism of bacteria down through a sediment that has ages that in this case, in the eastern tropical Pacific, goes from a modern and then down to the ocean crust here at 120 meter Sub-Seafloor, where the crust has an age of 37 million years, and that means the sediment was deposited 37 million years ago. And what you see in this case is that DIC, which indicates the amount of CO2 produced by mineralization of organic carbon, is not increasing with depth. Sulfate, if sulfate reduction were a dominant process there, it should be depleted. There is no indication of sulfate depletion. So what is the respiratory process that takes care of the net oxidation of organic carbon? Well, it turns out that this is metal reduction. And at the surface of the sediment, you have a deposition and burial of manganese oxide, of iron oxides. And you can see here from the production of reduced manganese ions in the pore water that it takes about 15 million years of microbial activity before they have depleted the manganese. And so we are working with complete different time scales than when we are used to, and it takes a long time before you start to think in these time scales. It doesn't mean that nothing is happening, because on a geological time scale, this is the main activity. This is what determines how much organic carbon will at the end be subducted and buried. So if you know the general sequence of the consumption of electron acceptors down through a deposited marine sediment, you will normally have oxygen respiration at the surface, then nitrate reduction, manganese reduction, iron reduction. And you can see here in this case, there's no methanogenesis, no sulfate reduction. There's manganese and iron reduction. How about the higher electron acceptors here? Well, we looked for that. When you core a sediment, you tend to lose the top few decimeters, and so oxygen was gone at the top. But nitrate penetrated about 2 meters. Then it was gone. Then you would say, okay, that's it for nitrate. Let's measure something else. Well, we decided to measure nitrate all the way down. And to our great astonishment, nitrate came back at the bottom. And there was also traces of oxygen at the bottom. And what turns out is that you really need to know the geology before you conclude about microbial processes. In this case, what happens is that there's flow of seawater down through the old crust that is tens of millions of years old. I'll just show an example of how this functions because one really needs to know it in order to understand what happens in the subsurface. And in this part of the Pacific, it has been noticed that there is channelized fluid flow through the crust. Has been known for a number of years. And it turns out that there are buried volcanoes. So when the crust passes over hotspots in the mantle, volcanoes shoot up. That's what you see on the Hawaiian Islands and the Galapagos Islands. And then the crust moves on and subducts again, and these volcanoes get buried in sediment. But it turned out that over those volcanoes, there is a conical depression in the seabed, and this is where the seawater goes in and out. And this has been published quite recently, that apparently through all these buried volcanoes there is a fluid flow, and the distance between them are a few hundred kilometers. The flow velocity has been estimated between 0.1 and 10 meters per year, and that means that the residence time of the seawater is between 20,000 and 2 million years. And the reason I stress this is that it means that over hundreds of thousands of years, microbial activity does not even use all the nitrate, and apparently also not all the oxygen that has come in. And that means that the rate of microbial respiration is extremely low. And what this shows is that there are apparently not hidden energy sources such as hydrogen production in the old crust that could supply these deeply buried communities with new energy. So what is the energy? Well, it's basically buried organic carbon. And how can there be anything left after 50 million years of burial? One of the things one needs to realize is the way that the degradation rate of organic carbon changes over time. Here's a compilation 20 years ago made by Jack Middleburg, where he compiled data for marine sediments, marine plankton, and what he found was that the degradation rate constant changed over time in a log-log linear fashion. So you have the logarithm of the degradation rate constant for organic carbon, and you have here the log of the age going from modern to 1 million years back, and you see there's quite a close log-log correlation. And what such a correlation means is that you never run out of organic carbon. You will always have trickles left, but rates go extremely low. So low that it's hard to imagine. We have tried to show such a change in the subsurface here in one case from a coastal sediment where we were able to measure the sulfate reduction rates down through layers that went from modern and to 8,000 years back. And you see with radio tracer technique to measure the current ongoing bacterial sulfate reduction rate here in picomole per cubic centimeter per day, we can show that there is a log-log linear correlation. That is, that the rate of degradation decreases with age and with depth according to a power law function. We have tried that even deeper on the shelf here off Peru. This is a compilation of a large number of data from different cores, surface cores, multi-cores, gravity cores, ODP cores, and the rates that we were able to measure directly with radio tracer dropped by 10 million fold from the surface and down to 100 meter depth in a log-log fashion. So down here at these rates, you hit the physical limit of what you can do with such a radio tracer technique. So if you go even deeper, in order to understand the real deep biosphere, you have to shift to modeling of pore water chemistry. And here's an example I'd like to dwell on a bit. Again, a core from the East Pacific. We see here the different geological times back to 17 million years ago, and here is the sulfate concentration in the pore water. Drops down, goes back up a little because seawater sulfate also comes up from below. And then we have used a model where we anticipate that the rate of organic carbon degradation changes in a log-log fashion with depth. And you can see that when you do a sensitivity analysis, if you had the double of our estimated rate, the sulfate curve would look like this, and half the rate here. So this sulfate reduction depth distribution is a very good estimate, especially in the central part. That is, if we are dealing with a pure diffusional system. Now, this, of course, is important that we are not also having advection. How do we know that? Well, we know because nature has done different experiments for us over time. Here's just one experiment, which was the shift from 100,000 years of glacial period, which ended some 10,000 years ago. And we're now in an interglacial, and what happened at the end of the glaciation was that all the fresh water bound in the polar ice caps melted, and then the ocean was diluted from a chloride concentration of 665 millimolar to 655 millimolar. And if this were then a diffusional signal penetrating into the seabed, one can model how deep it should have penetrated after 10,000 years. And the answer is 25 meters, and that's what you see, which shows that at least on this time scale, this is a purely diffusional system. So we have, out of this, determined the rate of sulfate reduction. We have here, by total cell counts or by fish counts, the total number of intact prokaryotic cells. And based on that, one can try to calculate then what is the rate of metabolism per cell in the subsurface. Now, in this estimate that I'll do, I'm assuming that 10% of the cells in this zone, which is dominated by sulfate reduction, are sulfate reducers. That is what we have found by studies in a number of different marine systems that are more active. And we know that the sulfate reduction at that depth, mid-depth, is 0.01 picomole per cubic centimeter per day. And if we assume that 10% of the cells are sulfate reducers, we can calculate the cellular rate of sulfate reduction as a mean rate, 10 to the minus 4 femtomole sulfate per cell per day. What does this imply? It implies that one cell is respiring 1 sulfate ion per second, which, when you start to think of it, is extremely little. And this is the entire carbon mineralization and energy flow available for the community, since sulfate is carrying 8 electrons in the reduction. It means that as a mean value, each cell has 1 electron available per second. Now, it's difficult, we cannot really relate that to something we know, but if you try to think of, for example, the growth rate of cells. We know from pure cultures how fast they grow. Here is a pure culture of sulfate reducers growing at, in marine systems under low temperature, and a growth yield on acetate, which is the dominant carbon source in the subsurface, is 4 grams dry weight per mole. Now, the growth yield tells us the coupling between the rate of metabolism and the time it takes before a cell can divide. And doubling times with such a growth yield and rate is 1 day to 1 week. Now, in the deep sub-seafloor, we had this cellular rate of metabolism. And what if they have the same growth yield? Well, then we can calculate that they can double once every 300 to 3,000 years as a mean value. Now, you can say this is not reasonable. 99% of them are not active, and only 1% are really doing something, and then you have much higher cellular rate, which is fine with me, but then you have to explain those 99%. How do they manage with a 100-fold lower energy flux than this to maintain apparently vegetative cells that are not spores, that have ribosomes, etc.? So my intuitive guess is that the rates of energy metabolism per cell may not be all that much different from this mean value, but this is just intuition, and we need to go in now with methods where we can analyze the metabolic rate of individual cells in subsurface samples. And we're working on that, doing long-term incubation experiments with tracer-labeled substrates, using then the nanosims to go in and analyze individual cells and then combine it with fluorescence in situ hybridization to know roughly where do they belong phylogenetically, and then to see which are metabolically active with the substrates that we offer them. And this is too early for me to show results that I really would be proud to present. But it is ongoing at the moment. So when you come up with such numbers, of course, you wonder, where's the flaw? Maybe there are other energy sources that we haven't thought of, and that geochemists will someday discover. And one such possibility could be natural radioisotopes. So there are very good data on the distribution of natural radioisotopes in the seabed. These are mainly thorium-232, uranium-238, potassium-40. And what is special about these radioisotopes and their high energy radiation is that the radiation splits water molecules. This radiolysis produces dihydrogen, which is a wonderful energy source, and then it produces different oxidized species, hydroxyl radicals, peroxide, that means that you have both an oxidant and a reductant available based on radiolysis. Just to give an example, with the potassium-40, it is very abundant, amazingly abundant. 0.1 per mil of all natural potassium is radioactive, highly radiating, with beta and gamma ionizing radiation. The half-life is 1.3 billion years, fortunately. So in each of us, there are about 4,000 disintegrations per second. So you're all radiating high energy gamma radiation all the time. But question is, could this support microbial life in the subsurface? Well, here's one example by the group of Steve Dond from the Eastern Pacific. And you see here the hydrogen yield is in the order of 0.003 picomole per cubic centimeter per day. And if you look at the level of sulfate reduction that we have modeled, you have here 0.001, which is about the level of sulfate reduction that this radiolysis could support. So it's still below that level here, but it's maybe only an order of magnitude lower. And there are, in fact, areas of the seabed where from theoretical calculations, the radiolysis would be the more important energy source than the buried organic carbon. And such ecosystems have been studied recently, but mostly in terrestrial systems with much higher radioactivity. So the last thing I'd like to touch upon is then, if we have the majority of all prokaryotes down there, are they an isolated community? Are they just a subset of what was originally buried at the sediment surface? There is a recent high throughput sequence study by Jan Biddle and co-workers that indicate that the total 60S diversity of archaea and bacteria is not decreasing with depth in the seabed. But all these sequencing efforts, of course, are not complete. They are still only comprising a part of the total diversity. Another interesting question is there genetic exchange between the subsurface and the surface world, or has this subsurface been isolated for tens of millions of years? And I'll do just a simple calculation to provide an argument in that discussion, and that is if we have transport by molecular diffusion, and this is what we can test by these geological experiments that nature has done, then we can calculate the molecular diffusion of ions in the pore water, and they tend to have diffusion coefficients of about 10 to the minus 9 meters square per second. And you can calculate the diffusion time, the mean diffusion time over different distances. And the diffusion time over about 250 meters, which is a mid-depth of the deep biosphere, is about 1 million years for ions. What with bacteria that are flagellated, that can move, but presumably have no gradient clue? If they live at a depth of 250 meters, there can hardly be such a clue. Well, there was a recent publication by Tom Fenchel that has shown that the effective diffusion coefficient of swimming bacteria without a chemical clue is about 10 to the minus 9 meters squared per second, which again would mean if they're swimming, the mean time it would take for an exchange of cells would be 1 million years. Now, for a few cells, it could be quite a bit shorter, but it does give an indication of the order of magnitude of time it takes for an exchange. That is if they're swimming. If they're not swimming, the diffusion coefficient is 3 orders of magnitude lower, which means that even an exchange down to 10 meters would take 3 million years as a mean value. So that means at a depth of 250 meters, there would basically be no exchange over tens of millions of years. So the question is, do they swim? Well, again, I know this is very crude, but I just concluded that they have about 1 electron available per cell per second, and a flagellar motor takes about 1,200 protons for 1 revolution, and a normal swimming speed 10 to the 4 to 10 to the 5 protons per second, which means that swimming is, this mean consideration, certainly not possible. And well, I'll leave it at that. There has been a study, metagenomic analysis, also hunting for motility genes, and you hardly find any in the subsurface, but that is just one indication. So, at the end, I would like you to remember that most of the prokaryotic cells on Earth are in the deep subsurface, and that these organisms must be adapted to extremely low energy flux. When you look at these doubling times, you would say there is basically no growth, and I would also conclude that growth is not the main parameter in the subsurface. You may have organisms that are lasting for thousands of years and that still function at extremely low energy flux. Growth and doubling is not a prerequisite for life. And we have to think in new terms. We organized an international workshop 2007 where we invited many people with different backgrounds in energetics, biophysics, microbiology, and had them discuss and think about this. And I think many came with much skepticism. One thing we got out of the meeting was that they got convinced that the data are of good quality and that here is a problem that is very interesting and for which we have no possibility currently to extrapolate our knowledge of the physiology of organisms to this type of environment where most of the cells are existing. And so we really need to start to study it. And I would postulate here at the end that the low energy limit for life is really much lower than we have previously thought, and it seems consistently in a number of different environments that it lies somewhere in the range of 10-fold below what is generally considered maintenance metabolism in anaerobic cells. So with that, thank you very much for listening. I'd like to thank those with whom I've been collaborating on this and exchanging ideas. And it's exciting out there when there are gas hydrates and high sulfide concentrations. It's not undangerous. So thank you very much for listening.
Happy to answer questions.
So the 85%, are they, you said you have 50% of the DNA sequences homologous to known genes, but 85% of the sequences were not related to anything that on the surface. Are they related to each other, I'm guessing? The 85%?
The details of that I cannot tell you because I have not really seen the raw data. This was work by Jen Biddle, and so I know what she has published, but I have not seen the original database of sequences. Okay. Please.
Do the fish measurements give you any idea of ribosome content of the cells?
Yes, there are certain proportionality between the fluorescence intensity and the number of ribosomes. In this case, we were using cardfish, which is a way of enhancing the signal. But I know that the group of Jen Biddle has used fish without this enhancement, and they have been able to pick up the fluorescence signal. And it's difficult to convert that into a number of ribosomes, but some hundreds of ribosomes in a cell, I would say, is required for getting such a signal.
Has anybody actually taken one of those sequences with a known enzyme from down there, from these slow-growing organisms, made it in E. coli and measured any kind of activity?
No.
Or is it a normal quality huge enzyme, or is it a very, very slow enzyme?
We have no idea. Yeah. We have very little idea of how this works. How obvious the enzyme kinetics may have nothing to do with the rate of uptake of substrate molecules in the cell. It's all controlled by external factors around the cell, and we don't know what is the capacity of the cell once it would have plenty of substrate. If you add substrate to these subsurface samples, you increase immediately microbial activity by 10 to 100-fold, so the capacity is much higher than what you see there. You could also ask, why don't you isolate these organisms and study them in pure culture? And that is also being done. You can isolate expected physiologies out of the sediments. I'm afraid that in many cases, if you can get things to grow in the lab within a few years, you have the wrong organisms. And I think we really need to develop techniques that are independent of growth and cultivation. And such techniques are becoming available rapidly, and so this can be done, but we're just at the beginning of that.
I'm sure you've given thought to the impact on evolution, right? So you have very, very slow-growing organisms, and also you have a strong gamma ray flux, presumably. Can a non-dividing organism evolve? Is there a selection over 1,000 years of survivors in the absence of replication?
This is a very good question, and then there's no answer to that. Some people have speculated, is evolution extremely slow if you have such low growth? On the other hand, maybe there are more mutations per division because of the radiation that is more dominant over the lifetime of a cell. We were discussing this morning the mutations that take place in spores, and what does that mean? How much DNA repair is required by a germinating spore? You could ask for a dividing cell, or just a cell existing there over 1,000 years. How much DNA repair must it do? How many mutations may there be over that time? Is there a lateral gene transfer in the subsurface? How much energy is spent on DNA repair? Are there viruses? Does that play any role? Are there bacteriophages? Yes, is that an energy cost? There are so many interesting questions, and really, this is a new research field in microbiology. I think we are gradually realizing that yes, this is real world. These numbers are not artifacts. There are properties of cells and their life conditions down there that are so different from anything that has been studied in pure culture so far, and we need good biophysicists and molecular biologists and others to go into this now. And this is my hope by giving such a talk here. I'm embarrassed to stand here and tell you stories for which we have no answers. On the other hand, I think here is a very interesting research field, and I think the technology, the ideas, the methods are now becoming available very rapidly to start to address those obvious questions.
The diversity in Arcadia, is that over multiple depths, or was it one step? And it was over multiple depths?
The one I showed was over multiple depths, and it also included hydrothermal systems in the deep sea and such. So it was a hodgepodge of different environments.
And did you see any correlation with the depths to the diversity, or was the diversity sort of seen across all depths?
It's too early to say. There's one large-scale sequencing effort made in the subsurface that was published. No, it's not even published yet, I think. With 200 base pair sequences, 454 sequencing, 500,000 sequences in that set, and you cannot really compare it to anything else. There is one such data set for a hydrothermal system in the deep sea, and I guess that is what is available at the moment. At least that's what has been published so far. And so I think within a few years we will have some answers to that, but it's only starting now. Yes, please.
Has anybody seen any hints of viruses there?
Yes, by simple microscopy, you can detect viruses. This Jack Middleburg from the University of Copenhagen has studied that. But if you are dealing with a system where cells do not grow, then to get to functional understanding of these viruses is difficult.
But they are seen in some of the set.
Yes. Mm-hmm.
Are there any traces of the viral genes in any of the genomes that you've managed data?
That's a very good question, and again, this 454 sequencing effort, of course, is for 16S genes, and the other I don't know. So since such a large part of the genes are unknown, well, you have the majority is simply unknown.
It's like kind of a naive question, but if you're working with a system that's so different from everything else, with all the metagenomic sequencing efforts, how do you go about figuring out what they are if so many of the approaches for metagenomics rely on comparison with things that we already know or can sequence? Where do you see that going when you're continuing to do sequence if you can't figure out more about the organisms with culture or other kinds of that? I'm not sure what I'm asking.
I'm not sure about the question, but obviously metagenomic sequencing in the subsurface is one way to go, and that is happening now. One main limitation in that, I think, is to convince people that this is an interesting field they should go into. Many shy back because it's much easier to work with fast-growing organisms with a well-defined genetic system and where you can predict better what you will learn. So.
If there are no further questions, I'd like to thank Bo for a very provocative talk about